One of the interests of my lab is to look at the glycosylation of receptors. And once these receptors are expressed on the surface, the question is, what are they doing? What is the major biological function or significance of these glycosylated receptors or the glycosylation? And so one of the things that we looked at is this toll-like receptors. And the toll-like receptors are sensors that sense bacterial pathogens. And these are expressed on the innate immune cells, the macrophage dendritic cells. And this sensing of the bacterial pathogens will initiate a signaling cascade that will activate the NF-kappa B to produce pro-inflammatory cytokines, so you get these inflammatory response. So one of the things I like to acknowledge my former PhD student, Samar Abdulkahelik, who did most of the work here in terms of this particular novel signaling paradigm that regulates toll-like receptors. And so basically, what we find is that this particular signaling paradigm also regulates not only the cell surface toll-like receptor 4, TLR2, but also intracellular TLR7 and 9. Intracellular TLR7 and 9 are the nucleic acid sensing receptors that are within the cell, within the intracellular compartment. So basically, the central process here is that there is a GPCR, namely the neuraminidin B, New1 matrix metalloprotease 9, which are complex together at the ectodomain of these receptors, whether it's the cell surface or intracellular TLRs. And they're both expressed at the naive as well as ligand-stimulated macrophages. So just to give you a little bit of background in terms of the structure, this is the toll-like receptors are have this horseshoe-type structure. And as you can see here, they are highly glycosylated. And these glycosylation sites seem to be randomly displaced throughout the ectodomain of the toll-like receptors. There are sections of the toll-like receptors, for example, in this region here, which are called the positive patch. This is where ligand binds. For example, if it's the LPS or any of these other TLR ligands, they seem to bind outside of the glycosylation sites. And there are several of these sites where the ligand binds. There's a transmembrane, and then there is a region here called the TIR, which is the toll interleukin receptor domain. So basically, the toll-like receptors are the frontline pattern recognition receptors of the innate immune system, and they play a very critical role in mounting an immune response, whether it's going to be an inflammation, but it could also be involved in autoimmune or chronic inflammation. So these play a very critical role. So the TLRs recognize not only the microbial pathogens, which are called the pathogen-associated molecular patterns, or PAMs, but also the intracellular toll-like receptors recognize the nucleic acid. Mainly, for example, when you have infections with viral, then these particular TLRs react to them and mount an immune response. So when they did the crystal structure, reveals that these toll-like receptors, whether they're cell surface or intracellular, they're highly glycosylated, mainly in the ectodomain. In particular, when you look at the crystal structure for the TLR3, which is an intracellular toll-like receptor, they have potentially about 15 N-glycosylation sites. And this is all depending on the particular TLR that's in question. So the mystery here behind all of this in terms of the glycosylation is how TLR activation is regulated. So when ligand binds to the TLR, it's well known that these toll-like receptors have to dimerize. So the dimerization process here facilitates the signaling adaptive proteins. One is called the MyD88, and you get the downstream signaling that activates NF-kappa B, which is a transcriptional factor, which translocates from the cytoplasm to the nucleus and turns on the promoter to produce these pro-inflammatory cytokines. So the basic question is, what is the role of TLR glycosylation in this ligand-induced TLR activation? We initially looked at this by accident, and one of the things that we were looking at, for example, in our earlier studies, looking at this T. cruzi, which is a parasite causes Chagas disease. And T. cruzi here produces this enzyme called transialidase. The sialidase cleaves sialic acid and transfers to its mucin membrane on the parasite. So we were able to get a recombinant transialidase from this T. cruzi, and when we added this to macrophages, we were able to get NF-kappa B activation and pro-inflammatory cytokine production. So basically what happened here is that this transialidase has substrate specificity for sialic acid, and in particular the alpha-2,3 sialic acid residue, which is the terminal sialic acid on the glycosylation. So this cleavage or hydrolysis of the alpha-2,3 sialo residues enables the removal of steric hindrance. And you get the receptor association, so you get the dimerization, as you see here, mediate complexing at the TR domain and downstream signaling to activate NF-kappa B. We were also looking at Streptococcus pneumonia neuraminidase. We found that this has very similar substrate specificity for alpha-2,3 Cl residue, and we're able to get the very similar pattern of reaction. So the very fundamental question is that, okay, here we have ligand binding to the TLR. An example is lipopolysaccharide, which is an endotoxin that binds to TLR4. And we get mediated receptor dimerization, mediated 88 recruitment, and activation. So the question is, ligand binding to the TLR, is there a mammalian sialidase very equivalent to what we see in T. cruzi or this neuraminidase that cleaves the alpha-2,3 Cl residues to facilitate the dimerization process? So we looked at this process here in terms of what may be happening. We published a paper in Cellular Signaling that we found that neuraminidase 1, which is a mammalian enzyme, it's a lysosomal enzyme, but we also find that this particular neuraminidase 1 is complex to the active domain of the TLR. And this complex here will facilitate the desialidation of the alpha-2,3 Cl residues, which causes dimerization and signaling. So in order to look at this, work by Hennick and Przensky and Stamatis looked at the actual structure of the neuraminidase 1. They found that neuraminidase 1 has to be complex with cathepsin A. This is a protective protein enzyme. It's necessary for the NEU1 to be complex to cathepsin A in order to become activated. Cathepsin A does not have to be activated in its enzymatic activity, but this complex here is essential for NEU1 activation. What keeps the NEU1 cathepsin A complex being activated here is that we have this elastin binding protein shown here. So the issue here is if it's an elastin, then maybe there is a matrix metal protease that has elastase activity that will cleave this, and as this removed, then you get this activity of neuraminidase 1. So one of the questions here is that if we had macrophages in culture and we surrounded these live macrophages with a substrate for neuraminidase called formunana, we add purified elastase enzyme, we're able to get this fluorescence surrounding these live cells. And this gives us an indication that this elastase has a capability to remove elastin binding protein to activate NEU1. NEU1 activation here cleaves the forminata into its byproduct that gives you the blue fluorescence. Elastase alone in the substrate here does not have any activity. We went ahead and asked the question here, well, one of the matrix metal protease that has a lasting activity is matrix metal protease 9. So we were able to get this MMP9 inhibitor, which is very specific for inhibiting MMP9, and we used this live cell Cialidase assay in order to test the activity. We used a negative control, which is MMP3 inhibitor, and in a dose dependency, we find that the MMP9 inhibitor is dose dependently inhibits this Cialidase activity as triggered by the LPS binding. So LPS binding to the toll-like receptor causes this MMP9 activity that cleaves the elastin binding protein, and you get mu1 activity. And we're able to show here in this mean fluorescent activity that MMP9 has an IC50 with 0.1 micromolar compared to the other one, which is very high potency. We went ahead and using the BMA macrophage, and we were able to show that the MMP9 inhibits LPS-induced phosphorylated NF-kappa B. And we did this Western blots here, where we're able to isolate nuclear lysates. We used a controlling peptide called phosphorylated NF-kappa B blocking peptide in order to show the specificity of this blocking. And we used the MCM2, which is a housekeeping protein within the lysate. So we were able to look at the densitometry of these bands here in a ratio to showing that adding LPS to these macrophage cells and removing these nuclear lysates, we're able to show a decrease in NF-kappa B activity. We also looked at the I-kappa B. I-kappa B in the cytoplasm is complex to NF-kappa B, and it prevents NF-kappa B from being activated. And it goes through a series of activation processes where the I-kappa B becomes phosphorylated. Once it becomes phosphorylated, it degrades. And so we're able to show here that LPS here degrades the I-kappa B in the cytoplasmic lysate, but when you add MMP9 or Tamiflu, we're able to prevent that. So basically, what we're looking at also here, just to confirm these results in a quantitative measure, we use this raw blue macrophage cell lines, which is a promoter cell line that is highly dependent on the SEEP enzyme called secreted embryonic alkaline phosphatase. And it's highly dependent on NF-kappa B binding to its promoter that activates the SEEP gene. And this SEEP gene is translated to produce the alkaline phosphate, which is secreted into the cytoplasm. So basically what we looked at here is LPS binding to the raw blue cells causes the dimerization, MyD88, downstream signaling. You get I-kappa B being phosphorylated, it gets degraded, and NF-kappa B gets translocated to the nucleus, activating SEEP. So this is sort of an assay system, very simple, beautiful assay system in order to measure a quantitative measure for NF-kappa B. SEEP activity being secreted is highly dependent on NF-kappa B activity. So when we looked at this in terms of MMP9 inhibitor and Tamiflu inhibitor, we're able to get relative SEEP activity in fold decrease quite significantly. For example, MMP9 here at IC50 is 0.1 micromolar versus Tamiflu about 2 micromolar. We used a positive control here called CAPE, which is caffeine acid phenylethyl esterase. And it's a potent specific inhibitor of this NF-kappa B. So we used this as a positive control. We further on looked at for example, at the genetic level, where we did lentiviral transfection of the sRNA into these round blue cells, and we did Western blots, and we find that this sRNA knockdown of the MMP9 was very specific in knocking down the activated isoform of MMP9, which is 88 kilodalton. It had very little effect on the pro-MMP9, which is 105 kilodalton. And then when we looked at the LPS stimulation here, we find that LPS was unable to get NF-kappa B activation as a result of sRNA MMP9 knockdown. We also looked at co-localization, confocal co-localization. We find that MMP9 co-localizes with TLR4, as shown in this overlay. We get about 45%, and when we add, this is naive cells, so this is already expressed on the cell surface without even ligand binding. When we add LPS at different time points here, we still get this co-localization. And this is confirmed by co-immunoprecipitation experiments shown here. We IP-TLR4, and we can actually blot for with MMP9, and it comes down, whether it's in the media, unstimulated, or stimulated with LPS. I guess the other question here is that if MMP9 plays a critical role in the activation of removing lasting binding protein, so it should be binding or forming a complex with the NU1. And so we did this NU1 co-localization with the MMP9, and as you can see here, without LPS, it co-localizes at the naive, unstimulated cells. And then when you stimulate these cells, we still get co-localization, but it decreases as the time progresses. And this is all confirmed by co-immunoprecipitation experiments, as you see here. The other question is the GPCR. And this is really a quagmire in itself, because the amount of GPCRs out there, about 800, 1,000 of them. And most of the drugs in industry are using drugs that target the GPCRs. And so the very fundamental question is, do GPCR play a fundamental role in the mediating this NU1 matrix metabolic crosstalk, and does it transduce transactivation the toll-like receptors and causes cellular signaling? So basically what we find so far is that NU1 and MMP9 form a complex at the ectodomain of the TLR4. And the question is, do GPCRs do also complex to this signaling paradigm? And so what we did here, as you see, is that we used a variety of different GPCR agonists. An example is bombesin. Bombesin is a 14 amino acid peptide, which is derived from the European fire-bellied toad. And it causes this, it's a potent, it causes this quite a bit of reactivity in that. The human homologue of bombesin is the neuraminidin, or gaston-releasing protein. So when we add bombesin to these round blue cells, we get this, in this live cell Cialis activity, a blue color, which is the activating neuraminidase 1. It cleaves substrate, gives you the blue reaction. Bombesin plus Tamiflu blocks this activity. We also tried bradykinin. Bradykinin is a potent vasodilator, and this is part of the archonic inflammatory response. And the vasodilator here relaxes the smooth muscles in the vessels, and the blood pressure drops. And bradykinin is a GPCR receptor, so when we add bradykinin to these cells, we're able to get activity. This is blocked by Tamiflu. LPA, which is a lysophosphatidic acid, which is involved in lipid signaling. We find the same thing. Angiotensin, which is a vasoconstrictor, we also find the same. And this is all blocked by Tamiflu. So here we're able to do a GPCR agonist that binds to a particular GPCR. And this GPCR is able to activate MMP9. And there's quite a bit in the literature that the GPCR agonist can activate MMPs directly. So here is the signaling paradigm that activates MMP9. MMP9 removes elastin binding protein that activates NEU1, and it facilitates desalidation and dimerization and activity. We further looked at this activity part of the GPCR in terms of looking at NEU1 deficient primary macrophages, which were derived from genetically engineered mice. Here we have a NEU1 cathepsin A. Both are point mutation. They have the protein, but the enzymatic activity is knocked out. And when we looked at LPS, we get no activity, whether it's from Bombesin, LPS, or even the poly-IC, which is a TLR. Cathepsin knockdown, which we thought maybe the cathepsin A would have activity on this. When we did a cathepsin knockdown, whereas NEU1 is still active, we still get activity. We also looked at, for example, MMP9 siRNA knockdown in the macrophages, looking at LPS, Bombesin, bradykinin, and LPA, and we were able to show that it was a significant reduction in this sialysis activity using MMP9 siRNA knockdown. Okay. So one of the other things here is that Bombesin induces NF-kappa B activation in raw blue cells, and here we use this GPCR activation, and this is another way of measuring a quantitative measure of NF-kappa B. And we find here that Bombesin here is able to activate CEAP activity, which is highly dependent on the activation of NF-kappa B. We can block this with Tamiflu at different dosage, or piperazine, which is an a broad-range inhibitor of GPCR, as well as the MMP9 inhibitor. And we also shown here that the Bombesin receptor neuromelanin B co-immunoprecipitates with the TLR in the cell lysates, as you can see here. And when you look at the NMBR GPCR, we find that there are different isoforms, and it's the 80 kilodalton isoform that is co-immunoprecipitated, as well as the 115. And here we show that NNBR immunoprecipitation is able to immunoprecipitate with MMP9 and vice versa. And we also looked at the Bombesin agonist in terms of reducing CEAP, and in terms of blocking MMP, MyD88 specific inhibitors. And we show here that this is the control, positive control with LPS, showing that if you do a MyD88 inhibitor, you can block this activity, whereas we can get the very similar response when you use the GPCR Bombesin. So MyD88 plays a critical role in the activation of the GPCR activation of NF-kappa B through the MyD88 recruitment to the dimerized TLR. Just to recap here, we published a paper in Cellular Signaling in terms of this whole matrix signaling paradigm with the NEU1 matrix metalloprotease 9 in association with the GPCR that regulates the intracellular toll-like receptor. It's an identical, striking signaling paradigm that regulates the intracellular toll-like receptors. which is phenomenal, is that the GPCR can also activate the intracellular toll, which has never been shown in the literature. And this is just showing the data here that we can do a co-localization as well as immunoprecipitation, showing that the new one, MMP9, co-localizes or immunoprecipitates with the intracellular toll. And I'll just move forward here. And the NMP9, the neuraminidin B, also is involved in this whole signaling paradigm. And lastly here is that when you look at the induced inflammatory cytokines in this raw blue cells, we can inhibit all of this with Tamiflu, MMP9 inhibitor, or even a broad-range GPCR inhibitor called BIM446. And here you can see here that looking at this MCP1 or TNF-alpha, these are pro-inflammatory cytokines, that you can get a very significant inhibition using these different inhibitors through this for the TLR7. We find identically similar kinds of response for the toll-like receptor TLR9. So in conclusion, what we find here, the new one plays a central role in mediating nucleic acid-induced intracellular toll, but not only for the intracellular toll, but also for the cell surface toll-like receptors. And it's the GPCR, NNBR, MMP9, U1 crosstalk that constitutes this novel cell surface as well as intracellular toll-like receptor signaling platform, which is essential for NF-kappa B activation and pro-inflammatory responses. And thank you very much for your attention. I'll entertain some questions.
Okay, thank you. Questions, please.
I have two questions. First
Okay.
one goes to the neuraminidase 1.
Yes.
I think you are working with the neuraminidase 1 in the endosomes, but it occurs in lysosomes and plasma membranes too.
Yes.
What is the pH profile where your enzyme is working? Is it at neutral or is it at acidic?
Well, when we did the live cell assay in terms of, for example, cell surface, these would be at the neutral.
At neutral?
Yes. Whereas when you do the intracellular compartments, which is the phagosome endosomes, they would be more acid.
And that's the same enzyme?
Same in what?
It's the same enzyme? The
Yes.
Same,
Yeah.
so it has a very broad pH profile?
Yeah, that's
This
right.
is proven?
Yeah.
Since there are 3 or 4 different neuraminases available in this area.
Yes, yes. Yeah, there are different isoforms of that, so the issue
Those are
is...
different genes.
Yeah.
Yeah. Mhm.
Yeah, that,
Okay.
I agree. This was a very challenging project because when you look at this, there are different isoforms, and so the issue is what isoform is involved in this particular
Exactly.
process,
Yeah.
which was quite a challenge. So when we did the blotting or even the immunoprecipitation, we had to do the full length and look at the whole signaling.
Now the second question is very interesting to the function of the oligosaccharide chains on your toll-like receptor. You have up to 15 different oligosaccharide chains, N-link. And now if you take them off, then you get the dimerization and the activity of the receptor. Do you have to remove them all by cellulase or only partially? Can you titrate it? So what's going on there?
Yeah, this is a good question in that you're correct. There's about, for example, like the toll-like receptor has about 9 N-glycosylations. The N-terminal is sialic acid. Using our assay system here in terms of our inhibition and so forth, we find that it's the alpha-2,3 sial residue. Now, do you need one or need all 9 of them in order to be desialidated? The very fundamental question you're asking is, has that been done? And the answer is no, we haven't done that. So all we know that it's critical to remove the sialyl residue. It's the alpha-2,3 that forms the steric hindrance. Removing that.
Yeah, but you have it, if you have 9, you have 9 times steric hindrance for dimerization.
Yeah.
If you remove only 1, I doubt it would work. Maybe you have to remove 4 or 5 or 6. You can titrate it. Can't you?
I think if anybody's interested in doing that, that kind, we haven't gone into that kind of
only a
titration.
question also of surface charge. Since ethoxysialic acid 9 has a negative surface charge, and 9 altogether is a lot of electrostatic potential.
Yeah, that's. I think we're interested more in the question is what is the functional role.
Okay. Yeah.
But as a glycobiologist or enzymatic, you're more interested in the questions you're asking, which is important.